Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Han Tan in July 2022. Han was a presenter at Science on Tap for the 2019 Maine Science Festival, where we focused on CRISPR, the gene editing technique. In this episode, we talk about Han's biological and genetics journey and how he's ended up doing really innovative and novel work on potato plants in Maine. I hope you enjoy our conversation as much as I did. Han, welcome to the Maine Science Podcast. I am delighted to have you here. You participated in the Maine Science Festival in one of, I think, our best sessions ever at Science on Tap about CRISPR. And what I tell people about that session is it was like the perfect science festival session because we had these amazing scientists. We had you and Michael Wiles from Jackson Laboratory talking about CRISPR. And at one point, I don't know if you remember this, but the bar became this back and forth conversation for the better part of half an hour to 40 minutes on the techniques and the ethics and what's it going to be. If I could have captured the essence of a Maine Science Festival event, it was that event. So besides the fact I loved the event, I was totally enamored with your presentation. So if I haven't thanked you enough for that consider this introduction a thank you for that.
Yeah, you're very welcome. And I would be more than happy to come speak at one of the events again at some point in the future. So
Excellent.
It was really fun. I was sort of relatively new assistant professor at the time. So it was really good exposure to get to know people in Maine. And, everyone always has amazing questions. So it was very easy to have that great conversation.
Yeah, I did remember the questions were really, really good. And some of it probably facilitated by the atmosphere of the bar, but I think a lot of it facilitated by how you and Michael had set up the talk. So before we circle back to the actual science that you do, which is, as I said, wicked cool, how did Maine get lucky enough to have you? I know that you, like me, were not born here and transplanted here. I always consider it a gift that I got to be able to live in Maine. I don't know if everybody feels that way, but I'm wondering how you got here, and maybe more importantly, what was your spark for science and biology?
It's a relatively long-winded, twisty sort of story, as most people who work in my field. So I'm a geneticist, and I've always liked biology. So I guess the best place to start is when I went to college. So I actually grew up in Malaysia, and I went to college in the US. So I went to Duke University in Durham, North Carolina. And at the time, I was, planning to major in biology because I really like biochemistry and I had some exposure in sort of genetics in some ways, but not in a very artificial sense, right? So when I arrived in college, I, like many students, were trying to figure out what do I want to do after within the first year of being in college. And so I was lucky enough to at first volunteer in a molecular biology lab working on plant genetics. But at the same time, I came in with a bunch of transfer credit. So I could actually finish my biology major very early, a year early, essentially. So I had time to also explore other passions of mine. So I really liked art at the time. So I was able to double major in visual arts. So I had a very confusing college career, I would say. I was getting pulled in two different directions. One of them is, hardcore genetics. I was mapping a gene for disease resistance in plants. By the other side, I was working on my sculpture project for my visual arts degree. So I tell this to my students whenever I teach genetics. I'm a very conflicted student at the time, and I applied to both graduate school and architecture school because I also really like architecture. But I ultimately decided that I liked science more. And then I did my PhD at WashU in St. Louis. It's a very biomedically related program, but I was able to find a lab that worked on plant epigenetics, and I worked on that topic. It was still a very interesting and important topic in genetics, and I still sort of follow the field a little bit. But after my PhD, I decided to go for a more applied field in plant biology. So I went to UC Davis and I was working on more breeding related things, but still using the plant model organism. And I postdoc there and start working on potato genetics by chance. And that's actually how I got the job at the University of Maine, plant genetics job there, because they were looking for geneticists as well as someone who worked on potato. And as you know, potato is a big, important crop here in the state of Maine. So that's sort of my long sort of story of.
Oh, that wasn't long. That was perfect. In fact, I've got questions for you. So first, you said epigenetics, and then you wanted to do something more applied. So could you explain what epigenetics is and what you may mean by not doing that anymore?
So epigenetics is a field within genetics. It's a realization when the DNA molecule was discovered, and studies have shown that DNA is the sort of the biological material that allows for heredity, right? When you have a mutation on DNA, you can inherit it and pass it on to the next generation. And that change in sequence is what's typically associated with whatever trait you're interested in. So that's what we call Mendelian genetics, which credits Mendel. But as people were studying some of the more interesting aspects of genetics, so you can think of epigenetics as a way of changing an organism's traits without changing the DNA sequence itself, because DNA itself is packaged by lots of proteins, and DNA itself can be modified without changing the sequence context. You can methylate DNA, for instance. That's what I was working on. It's just a small chemical change on the sequence itself. And when I was a PhD student, I was trying to understand how these different marks on the DNA, be it on the protein that wraps DNA or the DNA methylation itself, is changing within the plant. And so that sort of is a way of regulating the expression of the gene or the DNA itself without changing the actual sequence. So that is sort of the way you can think about epigenetics.
And you said you were a visual artist, double major as well. Has your background or interest in that made its way into your science world from the perspective of having a better understanding of visually showing people what you're working on?
I think so, but I was able to do a number of graphic design courses as part of my art degree. And I feel those, and photography as well. So those classes really primed me for doing microscopy work. The idea of exposure, the idea of light, the idea of composition. And as well as in science, when we present our work, we have to put PowerPoints together. We have to make posters. We have to make figures that are self-explanatory in our publications. So all of that added up together with sort of my, at least in my mind, my artistic side of things can still be expressed. And I do enjoy doing that as well. And in fact, even when I'm here as a professor at the university, I've actually designed 2 logos for 2 big grants. That's one is in the USDA grant and one is for the NSF grant. So I've designed logos for these different consortiums as well as institutes that I've been involved in. So that has come in handy because you always need some sort of visual representation. And that is, to me, is an important sort of branding type of things that we do as scientists that is sometimes ignored, right? And we have to be a little better at doing that.
I think that's really neat. When you were at UC Davis, did you expect to get into the world of potatoes? I mean, I don't think of California and Davis in particular as a potato stronghold.
No,
was it just a convenient, like a lucky break with the plants that you were working on?
I guess yes and no. So when I was in Davis, we were working on a way to manipulate the number of chromosomes in plant species. And one of the ways is actually using a DNA related protein. It's a histone protein that I was working on in the lab. But it so happens that when I started my postdoc, we still know very little about the mechanisms in which this mechanism occurs. So in the lab, we were able to change a single gene. So I was working on a technique called haploinduction. Essentially, you are reducing the number of chromosomes from the parent, and in the offspring, you will have half the number of chromosomes in the parent. So that's a very useful tool that plant breeders use and have been using since the 60s, actually. But in crops such as maize or potato, the mechanism for that cross is completely unknown. So it was a completely black box. And the lab that I joined had figured out a way to manipulate that by using a specific gene. So our interest in potatoes stems from the fact that when I was working on the project using the plant model, so this is now using Arabidopsis thaliana, so it's like the mouse of the plant world. We have a lot of genetic tools, a lot of techniques that we can use to make changes to the genome that we can then study. We were trying to see if it's the same mechanism in potato. So we were able to sort of try to get the information from our model system. And by using the potato as the proxy, we were trying to figure out if there's any connections between the induction process of potato compared to the one in Arabidopsis. And that's actually what my lab is currently still working on. We still don't quite know what's actually happening. To me, really fascinating.
When we moved to Maine, when I finally got to go back to paid work, my first job was a grant and kind of analysis of, can you grow enough potatoes to pull enough starch to make bioplastics? Kind of pie in the sky thing, really interesting. But I learned a whole bunch about potato farming, not the potatoes themselves. So I learned very quickly after moving here that potatoes were a hugely important historical and current crop in Maine. And, oftentimes lobsters certainly are at the forefront that everybody thinks about, but it was a great introduction into how vital and important potatoes are to Maine in many, many ways. Since then, back in the dark ages of 2004 or 5 or whenever it was when I started that, maybe a little bit later, I've always kind of kept tabs on potato world of Maine in a very broad way. And I know there's been a lot of really interesting genetic work to breed more resilient potatoes for all sorts of different things, or just different types of potatoes that are good for things. So what I'm curious about, before I let you really dive into your science with the potato and what you're doing right now, is the reason that potatoes, and I don't remember what other crop you said, is a black box. Is it because it's so complicated, or is it because the first plants or crops that people figured out were the ones that are larger and considerably more vital to the world in terms of production?
So in the plant breeding world, one of the most successful crops, I would argue, is maize or corn. So that one of the cornerstones of agriculture in a lot of countries, including the US. So early on, a lot of research has already gone into trying to develop better maize varieties. And one of the ways that they were able to do that was a method called hybrid breeding. So essentially, it's a way to make crosses from two different parents, and you create what we call these F1 hybrids. And because of the genetics that is involved, you get these huge increases in yield, in biomass, in potential for this crop. You can't do this in many crops. Maize is one of the few examples where this has been highly, highly successful. And one of the reasons why it has been successful is because of haploid induction. Because in order for you to get to the original parent, to make that cross, to make the hybrid seed, you need the two parents to be highly inbred, which means the parent itself has to have almost 100% similarity in the two sets of chromosomes that they carry. And when that happens, you get the maximum amount of this vigor that is associated with hybrids. Because of that, there's a method of inducing these haploids that can then become these inbred parents in maize. That technique relies on a number of these special lines that they call haploid inducers. And in the maize world, the base number of chromosomes for corn is already diploid, so that makes it a lot easier because you're only working with two sets of chromosomes in that sense. So because the haploid induction method in maize had worked so well, people basically wasn't really interested in understanding why. And it wasn't until I believe it was 2016 or so where they finally figured out what that gene that was involved in maize haploid induction was. Basically, it took over 50 years to get there. So potato, if I can circle back to potato, is a very different breeding system. the conventionally bred potato, the ones that you eat, the one that you have as French fries and tater tots and potato chips, they're all tetraploid. So the base number of chromosomes for those are 4 sets of chromosomes, which means that the breeding process is a little more complicated. And if I can backtrack a little, around the same time that people were trying to understand haploinduction, they were making crosses in wheat, in oat, in maize, in potato, in tobacco. And what's really cool is this method has been known, as I said, since the 50s and 60s. So it's not like an unusual technique, but in the potato system, when people tried to do that, they had a really hard time getting diploid potato. So if you reduce the tetraploid potato, you get diploid potato. And there was a big push about 20 years ago to try to get potato to be more like maize. But because of the tools available, we didn't have a genome sequence, we didn't have all the genetics, we didn't have CRISPR, we didn't have the transgenic. So there's a lot of things that had to happen. We didn't have the sequencing tool that was cheap enough, et cetera, et cetera. So basically that effort sort of went away because there weren't any successful potato that were bred as a diploid.
Are you working on diploid potatoes now?
Yes. Yeah.
So the stuff you're working on now, you're able to do because of the extraordinary advances that have happened almost since you've been an undergrad or a little bit past that. I had someone tell me once that bioscience and biology and genetics is kind of like the new rocket science from 50 or 60 years ago. Like the advances that were made in engineering and rocketry that just were leaps and bounds, has built up what many would call the society of the last 30 years. Those types of innovations are where we're at now. So you're able to do this work because of the innovations that have happened. And it sounds like the tools have caught up with the science questions, which leads to what you're working on isn't just figuring out the potato mechanism, right? Aren't you also working on specific types of breeds and specific types of hybrids? And I'm going to stop and let you explain all the cool stuff you're doing and ask you only if you can also, in the midst of this, explain CRISPR in a way that will hopefully make sense if people haven't heard of it before. No small task there, huh? Have at
it. I will try. So basically, as I said, not all crops are bred equal, I guess. The tools available for potato breeding is very different than the tools available for tomato breeding. And the tools available for tomato breeding is very different for rice breeding or maize breeding, because you not only have the different types of species involved, so the domestic, the basal sort of species, and you have these big classifications of monocot versus a eudicot crop, et cetera, et cetera. So in a lot of sense, the way that I like to describe breeding is that the people in the maize tomato world are sort of much farther along. They're maybe at the SpaceX kind of situation, while we're maybe in the Apollo side of things, because the way that we've been breeding potato has not changed since the domestication of potato, which was like 8,000 years ago. Basically, people are just making these random crosses together and hoping that as you move along in your breeding population, you are improving potato incrementally. And we have done so successfully. And I like to show this chart of like yields of potato over time in the same time period as maize. Potato is actually doing really well in terms of the gains that we have, the genetic gains that we have. But I think it can be improved. Like it's comparable to maize, actually, even with our traditional method. But the thing with the potato breeding is that it takes 10 to 15 years to get a new variety that is acceptable to the industry as well as consumers. So it's the adoption of these new varieties that is very difficult, because there's not that many to choose from. And you need to develop one that can be grown basically from Florida to Maine, which is for the most part almost impossible. In most crops, you're breeding for specific regions, but the potato world is slightly different. You have to have a bigger adaptation. But I'm sort of running away from a question a little bit. But the reason why I said that, because by changing the ploidy of potato base number from tetraploid to diploid, which is what my lab is doing for the last 3 to 4 years, we hope to be able to then create more lines of potato to be tested at a faster pace. And we can also be able to fix certain things, because in the tetraploid setting, you cannot fix something because of the 4 sets of chromosomes. It's a lot easier to fix something when you have 2 sets of chromosomes, right? So instead of having 4 decks of cards, you have 2 decks of cards. It's more likely that you find the combination that you want when you have less complexity. So in a nutshell, we essentially trying again. So there's a big push, not just from the Maine breeding program, but from the breeding program from across the nation. So potato is really interesting in the sense that it's the last remaining public breeding program in the country. So there's potato breeding programs in North Carolina, in New York, in Maine, in Michigan, Wisconsin, Minnesota, Idaho, Washington, Oregon, North Dakota, Colorado, Texas. I might be missing a few, but it's the few publicly supported programs in the country. So most crops have gone to private companies, and you rely on, there is a few private companies in the US, and the only one here is PepsiCo. But most of the potato that are grown in the country are released by public programs like the one at the University of Maine. So that's also a characteristic of potato that's slightly different than other crops. But because of these advances in sequencing and our tools available to be able to get genotype information. So genotype information essentially is we sample, it's like when you go get a 23andMe, it's actually the exact same technology as 23andMe that we use in potato. So we get a snapshot of important regions on the genome. And because of the availability of the software, computation, technology, so it has to all come together in order to make it economical. And because we are all public programs, our funding isn't as consistent or as high as if you were working in a private company setting. So by pulling together our resources, my lab and others have started to develop these diploid potato lines that we can then use as these foundational pre-breeding lines that we can hopefully be able to get to the level that you can see in the tomato, maize, and the other sort of crops that have been bred in sort of the setting in which you can have faster turnover and faster adoption of these newer varieties.
So are you hoping your work and the other folks' works are, instead of taking 10 to 15 years, if you're able to get the diploid baseline and really understand the genetics, how fast do you think you could turn out new crops that deal with everything from consumer acceptance and ease of growth to, let's be honest, climate change and the impacts of the changing environment, and making sure that the potatoes themselves are resilient?
Yeah, that's an absolutely great question. I think we can maybe shorten it to even 5 years, I think, because I have a graduate student that's going to be starting in my lab this coming fall, and we are using these diploid potato in making monoploid. Essentially, we will try to go from a diploid crop to a true haploid. So this will be very close to what the maize people are doing. And they're able to then get 2 rounds of breeding cycle per year. So a lot of the maize, for instance, are grown in Hawaii, and then they grow it in the US. So they can continue to do the breeding. So that's my goal, is to be able to get a similar setup in which we can push along these. So waiting during the winter months, we can consolidate programs in greenhouses or in other places in the country. Like Florida, for instance, we have collaborators there, and you can grow potato pretty much in January in Florida. So there are definitely opportunities there that we're still developing, that we can fast track some of these processes to get enough diversity in our lines to then be able to test. So I think that's the key here, is that all the ties that I talked about with these different breeding programs, we share all our resources. We share personnel, we share germplasm. And because it's a public program, there's less restrictions in terms of sharing the genetics. If you're a company, when you acquire companies, you protect them and it's all proprietary. So we're working in a more open setting where we hope because of that, we can start to generate material that is acceptable across the different growing regions for potato. So then maybe perhaps we can be more targeted as opposed to trying to breed for one variety that can be grown in a wider geographic region. We can target it to grow in a more specific region, and as climate change or changes in the environment, disease changes, we can then very quickly respond to that by tapping all the wild relatives of potato, for instance, as well as all the other tools that we can use. And that might be a good segue for CRISPR. If you think about it, you can think of CRISPR as a way to very quickly generate changes to the DNA sequence itself at this very specific place. So you can basically target a change and be sure that for the most part, that's where you're targeting, and then detect the change later on. And that's really important because this is where all the diversity comes from, right? So when you have, if you think about a good example is the different COVID variants and subvariants. These are all changes to the nucleic acid code, right? So in order to do that specifically, it used to be quite difficult. You have to initiate it randomly, either by chemical treatments or by irradiating. So gamma rays, actually, I used gamma rays to meet mutants in plants before. But with CRISPR, if you know the place where you want to make the change, and this is where having a really good genome, a reference genome, is important too. So it's not just CRISPR, you have to know where to go. You need a roadmap to know this is the road that I want to change, right? So in order to do that, the way that CRISPR, even though it's discovered in bacterial world, the leap in that innovation is the fact that you can do it in any organism that has DNA. So hopefully that makes sense. So we can then target, say, if you know a gene, there's a gene that's really important for powdery mildew resistance. And if you change that gene, that sequence on that gene, you then will have resistance. And in that sense, traditionally, if you have tetraploid potato, you have 4 copies of that gene to change. And based on the studies that have come out over the past couple of years, it's almost impossible to make the same change on 4 copies because it's a lot of things to change, but it's a lot easier to do it if you have only 2 copies. So that's another benefit of why we're working on diploids, because you can very easily make changes without having to worry about tracking the different copies. And even though we have really good sequencing technology, you still can't always tell the 4 copies apart because they're so similar. It's a lot easier when you have 2 copies.
Yeah, I mean, it seems like by reducing the variability, you just reduce the chance for error. So
pretty much.
I feel like this is such a dopey question, but I just have to ask it. Is there a risk to reducing the number of chromosomes from 4 to 2 from the perspective of vibrancy or ability to withstand changes? Like, are you putting limitations on the new version of the plant that could actually have significant side effects? Or am I reading too much into it?
No, you're absolutely right. Evolutionarily speaking, the conventional potato have been existing as a tetraploid for a long time. And because of that, there is a lot of issues that's associated with reducing the number of chromosomes, because you can accumulate more deleterious mutations when you're at the tetraploid setting than if you're a diploid. So when we are doing this in the lab, that's a huge hurdle that we have to work with, because we are essentially going to be increasing the number of deleterious alleles when we decrease the genome by half. And that could have profound effects on the resulting diploid. So one of the reasons why it's so challenging, and that's why it wasn't as successful when they were trying to do this in potato years ago, was because of this. So they were observing a lot of issues. So when you have these regions of the genome that are segregating in the diploid population, you might encounter issues at later generations that you might not know of. But the good thing is the group that I'm working with are recognizing this as a problem. And what we're doing now is because of the long read sequencing technologies and also the fact that we are now much better at assembling genomes, we're actually creating full-length phase genomes from the initial population that we're working with. So then we can perhaps be able to capture some of the issues and try and circumvent it by sort of looking through our selected populations of these initial diploids to make sure that we are not inheriting these deleterious regions. And it also might help us target regions where we want to avoid, essentially. So if we know that this particular clone has this region that is really bad that we don't want to be carrying on in our breeding population, we can eliminate those early on. And we can develop markers that can help us do that. And in the potato world, there's typically 2 to 3 big regions of the genome that you might want to avoid based on what we know.
So you're basically able to get a genomic map for each iteration and make sure, as much as you know right now, that you're just picking the good parts.
Yeah. And then when we are recombining them by making these specific crosses, we can then be sure to only keep the individuals that have the good parts and avoid individuals that have the bad parts. And we have a way of doing that.
It's so cool. So I'm going to make you talk about 2 other things in shorter detail, if you don't mind. The first one is, I noticed you have a ton of undergraduates as alums of your lab. And I know you're still assistant professor, right?
I will be associate professor this fall.
Oh, even better. So forget that. So for people who don't know, the academic treadmill, such as it is that you're assistant until you get tenure and then you get awarded tenure. So congratulations. I knew you were around the time where hopefully you were up to tenure, which actually makes kind of my observation, I guess, which will lead into a question even cooler to me. You rely, from what I can tell, pretty heavily on undergraduates and master's level students for your research. And that is not a trivial thing when you're trying to make a name and establish your lab, because you have a ton of training that you have to do for those students, more so than graduate students or postdocs. So I'm curious, I assume you feel good about that because you have a very long list of students, but also how does that compare to the places that you've been? And I will admit that this is a leading question in the sense that a lot of other University of Maine professors that I've spoken with and some folks at the University of New England have made a point of talking about how important undergraduates are to their work. And I think it's a part of what is on offer at the University of Maine that people don't recognize is different than other places. So I guess I'm asking, has that been your experience as well?
Yeah, I totally agree with that sentiment. And sort of go back to your earlier question about my experiences with undergraduates. I actually have been working with undergrad since I was a grad student. So I was lucky enough to be able to mentor 2 undergrads when I was in graduate school. And then I was able to mentor, I think, at least 4 undergraduates as a postdoc. And that's what sort of led me to wanting to provide opportunities for undergraduates, even high school. I actually have a number of high school students as well that have come through the lab. And it's mostly because when I was a postdoc in Davis, I worked with a group there that basically was working with mostly underrepresented minorities, undergraduates within that group, and they were placed within specific labs. And I just had a really positive experience sort of, leading these students who didn't know anything about genetics to, helping them create CRISPR constructs to modify a chromosome. That has always been very, to me, like rewarding because you see them grow as a person. So when I started my lab at UMaine, I was able to also recruit a number of students that way, mostly because I teach genetics and I get a number of inquiries every semester after the class, typically students who are really interested in the topic and want to learn more about my research as well as getting some hands-on experience in my lab. So I've always tried to encourage that at the University of Maine because we have the honors college as well. I've also mentored or co-mentor a few and mentored 1 honor student in the lab, and that has always been a really positive experience. And of course, we have capstones as well. So I've also had a number of capstone students that have contributed to all the things that I talked about. And for the most part, I think it helps them understand whether A, they like science, or B, they don't like science. And I think I'm about 50-50 in terms of people who realize that maybe this is not for me. And that's completely okay. And I tell them that it's good that you're recognizing your limitations and your interests. And the people that are interested in science have gone into other graduate programs. I've had students who went to UCSD for a PhD program. I've had students working for Pfizer, students working for a lot of the plant biotech industry. So I think that gives them the leg up for their next chapter of their lives. So I feel that giving them the opportunity is important, because I was given the opportunity when I was an undergrad too.
I think that's a perfect synopsis of what makes a great faculty professor. From my experience as an observer, as someone who doesn't do it, I think it's really great. And my last question for you, which I will admit is totally ridiculous, is what's your favorite way to eat potatoes?
Absolutely French fries all the way.
No kidding. Okay.
I am a very big fan of French fries, I must say. And it's one of the few food that I've always liked as a kid. I don't know. I think there's so much variety in French fries, and there's so much, I don't know, it's very simple. It's just fries and you cook them well with a little bit of seasoning. And I really do enjoy it. It's satisfying. It's super convenient. You can eat it in your car. I don't know. I feel like it's the simple things in life.
You're not going to get an argument from me. I have yet to have a potato in any way that I don't like. And I think fries are one of the best ways to have it. So maybe now that you have tenure and you have a chance to breathe just a little bit, you can figure out a way to take your visual arts skills and team up with the art department and do some really cool potato molecular structure that can better describe to people who don't understand the way that we're talking about your work in a way. Because you don't have enough to do. You should also take on an art project like this, Han.
I know. I think at UMaine, they just renovated a really huge art space that has people in engineering, and I have yet to visit it, but I've heard about it from the tours that people have been taken to that space. So I think there might be some opportunities there. I just haven't. As you know, we wear many, many hats.
You wear too many hats, and I really do not mean to put another one on you.
But that would be fun. It could be a fun, I think, honors student project even. And I can see some connections there, or even doing some VR things in the potato field, or there's all kinds of stuff that we can do.
I mean, I'm mostly kidding, but I do think that there's a lot of opportunity to break down the silos between the arts and the sciences in ways that make both more inviting to the people who think they can't do one or the other.
Well, your question about the food actually had me thinking, and also that's something in the back of my mind the last couple of years. Food brings people together, and potato is found in all cuisine. And I think what we need to do, maybe the Maine Science Festival, would be to do a potato cooking contest, potato food. I don't know, some sort of.
Ooh, I love that. I'm going to get together with my team and see what we can figure out, because that would be really interesting.
Pretty cool, because you can get all the cultural differences, and then we could supply different potato varieties and, things like that. That might be a good way to bring people together and talk about something over comfort food.
I love this idea. I'm serious. I'll throw this to my team and we'll see what we can come up with. This will be really great.
Well, let me know if you want potatoes, because we might be able to get you some.
Yeah, that's already part of it in my head. You don't have to worry about that. Han, this has just been a joy. I really appreciate this. I especially appreciate your generosity with your time. How willing you are to talk about a really complicated genetic stuff. When I went to school, it was just biology. We didn't talk about genetics in any way beyond Mendeleev. So I really appreciate it. I think it made a lot of sense, and I hope that people enjoy this as much as I have.
Great. It was a pleasure talking to you, and thanks for inviting me and giving me this opportunity.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by subscribing on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is hosted by me, Kate Dickerson. This episode was edited and produced by Scott Loiselle. We receive financial support from Central Maine Power, production support from Miranda Bouchard, and social media support from Next Media. The Discover Maine theme was composed and performed by Nick Parker.